Every 90 minutes, someone dies from ALS. Would it shock you to know that some cases of this disease may be caused by a virus? Endogenous retroviruses, or ERVs, are a type of virus that resides within your DNA. They do have some benefits, like a role in normal human development, but as you age, these ERVs become silenced, so they're not active anymore. However, a situation could arise where these viruses become active again and can then wreak havoc on your DNA. A viral protein found in the brains of patients with ALS is the ERVK integrase enzyme, and to explain its function, I want you to imagine two ribbons. One of these ribbons is blue, and that's the viral DNA. The other is red, and that's human DNA. Scissors, or integrase, will cut the red ribbon and insert the blue ribbon into it, therefore inserting viral DNA into human DNA. Human DNA will then alert the body it has been harmed and activate a damage response to try and fix this problem. ALS, or Lou Gehrig's disease, is a fatal neurodegenerative condition. It is notably characterized by DNA damage in neurons, the cells in our brains. DNA damage leads to neuron death, which impacts the control of voluntary muscles, like your arms, legs, face, as motor neurons in the brain lose the ability to communicate with muscles in the body. So I ask, if ERVK integrase causes DNA damage, could a mouse model confirm that this viral protein leads to the onset and progression of ALS-like symptoms? My lab has already shown this in fruit flies. Now we move on to a preclinical model with mice, using a control group that does not express the viral integrase protein and a test group that does. So here integrase is on, doing its work damaging DNA. We'll run a series of behavioral tests on these mice to monitor their motor ability, which we expect will decline in the test mice compared to the controls, and continue this for 9 months or more. After, we will examine their brains with a microscope to confirm the expression of the viral protein and its association with DNA damage in brain cells. This may have started with a mouse, but by showing that a viral enzyme leads to motor dysfunction, a new world of therapeutic options is now available. Right now, there are 3 approved treatments for ALS. This research can allow us to repurpose antiviral drugs that are now used in cases like HIV and use them in ALS or other ERVK-associated diseases like cancer. By understanding the viruses within us, my project has the translational potential to provide a new model to investigate the association between viruses and neurodegenerative diseases, as it becomes a platform to screen new antiviral drugs. This could change the clinical outcome for patients and give them hope for tomorrow, where little exists today. Thank you.